that should give us, hopefully, enough data to answer these questions. The pilot project itself is a huge undertaking. It's led by Stephen Richards at Baylor, and they've already sequenced 25 new arthropod genomes. And each of those is being analyzed in depth by individual groups. But what I want to do today is provide a phylogenetic context for these new genomes so that we can start to observe patterns of evolution between these species. So with that in mind, we took the 27 newly sequenced genomes, combined them with 49 previously sequenced arthropod genomes, which gives us a data set of 76 arthropod species that spans 21 arthropod orders. And with these data, we'll be able to ascertain the relationships between these species and start to ask some questions about evolution between species. Now, I have time to address a few of these questions today. I'll start with inferring the phylogeny. And this is interesting in itself, but it also, the species tree serves as a scaffold for us to ask these other questions. So the first thing we need to do is figure out how the genes from our 76 species are related to one another. And this is a lot of work done by Rob Waterhouse at OrthoDB. And what I'm showing here is just the number of genes for each of our 76 species and a couple of outgroups for comparison, and their membership in certain orthology groups. And when Rob was done, he was able to give us about 38,000 gene families, orthogroups, for us to work with. So with so many species, 76 species, we weren't able to find any single copy orthologs among all of them. So when constructing the phylogeny, we had to take a different approach. And what we chose to do is construct a backbone phylogeny at the order level, and then construct 6 separate order trees of the 6 orders that had multiple species. So when we do this, we kind of had to change our perspective of what we were calling single copy orthologs. And in this case, what we wanted were gene families that were represented in each order by at least one species that was single copy. And when we use that criteria, we get 150 single copy groups at the order level that we can use to reconstruct the tree. And we went ahead and ran these genes through 2 alignment programs and 3 species tree methods. That'll give us 6 species trees that we can compare to one another to see if there's any disagreement among them. And I'll go ahead and just show one of those trees because for the most part, they agree with one another. And this is the tree from the pasta alignment of these genes and reconstruction with the coalescent method Astral. And I think this ordering pretty much agrees with the accepted relationships among these lineages. There's some lower bootstrap support in these sort of ancestral holometabolonodes, but the ordering among the lineages was the same through all 6 methods. Where we do see something interesting, though, is with the crustaceans. We recovered a monophyletic crustacean plate. And I just want to focus on the crustaceans for a minute, because it's kind of interesting. So I'm going to simplify the tree to the 3 crustacean orders, insects, and myriapods as an outgroup. So we recovered this monophyletic topology, and this actually differs from other studies that have recovered this paraphyletic ordering with the cladospheran sister to insects. So I wanted to kind of examine whether we can tell which one is correct, or even if we can tell which one is correct. So among these 4 lineages, there are actually 15 possible topologies, 3 of which are monophyletic. And with our 6 methods of species tree reconstruction, we observed 4 of them, none of them being the previously inferred paraphyletic ordering. So I kind of wanted to examine this trade-off. With 76 species, there were no single copy orthologs because there are so many species. I wanted to see if we lowered the number of species to increase the number of sequences we used, whether we got more agreement among methods. So I lowered the number of species from 22 orders to 17, and that gave me about 400 sequences. And when we do this, we actually recover 3 additional topologies, so even more disagreement among methods, and we still haven't recovered the previously inferred paraphyletic ordering. So I decreased the number of species again from 17 to 12. That gave us about 1,000 sequences to use. And still we get 2 more topologies, and we finally recover the previously inferred ordering, but still a lot of disagreement among methods. We do see one of the monophyletic ones sort of starting to be favored, and actually about half of our methods recovered monophyletic crustacean. So as to which one is correct, I don't think we can say at this point. Whether we maximize the number of species or the number of sequences, methods disagree with one another. Of the 15 possible topologies, we recovered 9 of them, and half the methods return a monophyletic crustaceans. So I don't think we can tell which one is correct based on these data. Moving forward, I had to pick one though, so I picked the one that maximized the number of species and used a coalescent species tree method. But I think it's important to keep the disagreement that I've talked about in mind before we make any conclusions about crustaceans in subsequent analyses. So with the backbone phylogeny in hand, we set out to construct the 6 multi-species order phylogenies. And to do that, we just took single copy orthologs within each order and did the same 6 species tree methods as before. And when we do that and sort of take those order trees and paste them onto the backbone tree, we get the full 76 species arthropod phylogeny. And again, we see a lot of agreement. In fact, for these 4 orders, all 6 methods gave the exact same topology. Within the other 2 orders, there was only a little bit of disagreement. Within the hymenopterans, the ordering of the parasitic wasps was contentious. And within the dipterans, the placement of the sandfly was sometimes next to the flies and sometimes next to the mosquitoes. But for such a big phylogeny, to see only a few contentious nodes, I think is pretty good. So with the species tree now, we can start to begin to observe patterns of genome evolution. And a natural first step, for me at least, is to look at rates of evolution. And what we want to look at are amino acid substitution rates and gene gain and loss rates. And we're hoping to find, to look for any variation among lineages in these rates. We want to see if any lineages are evolving exceptionally fast or slow. And then we want to compare the rates of these 2 events, because that might say something about what is causing any rate variation. So we want to compare these rates on a scale of absolute time. So we need to take our tree and scale it by time. And we just used a simple non-parametric method and some fossil calibrations to do so. And so this is the same topology as before and now scaled by time. So to get amino acid substitution rates per year, we just took the branch lengths from the unsmooth tree and divided them by the branch lengths in this time tree here. And to visualize this, what I did was I made a histogram of the rates, which I'm showing as a density plot in the lower left-hand corner. And I sort of color-coded the bins. So each lineage has a rate that fits into a bin with a color, and then I just took that color and colored the branches on the tree again. And what we can see here is that most of the rates seem to fall into 1 or 2 of the bins, the sort of blue and purple bins. And there isn't much, there is variation, there's a tail there, but it's mostly constant. There is a little bit of a speed up in sort of the ancestral holometabolite nodes. So if we want to sort of identify what are the causes of the variation we do see, we could take another type of mutational event, such as gene duplications and losses, and compare them to one another. And so because these 2 events have different underlying mechanisms, if they are correlated, like the rate changes are correlated, then it's something common to the organism that is changing, such as a life history trait like generation time or population size. And if the rates are uncorrelated with one another, then it's changes in the underlying mechanisms that are probably causing the differences. So I took the 38,000 gene families and calculated ancestral states for the gene counts and just counted the number of changes on each lineage. And then I divided that again by the time tree. So when I do that, now this is the gene gain and loss rate per year. We get the same sort of density plot and colored lineages. We see in the density plot, we see mostly low rates. They're all confined to mostly 1, the lowest bin. So when we look at the tree, it's just sort of a wash of teal. Most of the variation seems to happen within hymenopteron. So back to whether the rates of amino acid substitution and gene gain and loss are correlated. If we put these 2 trees next to each other, it doesn't appear that they are. So when we see changes on one lineages on one side, so on the right is a substitution tree now, and on the left is the gene gain and loss tree. And when we see changes on one branch on one, we're not seeing corresponding changes on the other. So this might imply that the causes of the variation of these types of changes are due to the changes in the underlying mechanism and not something common to the organism. So now I want to shift from rates to actual families that have been changing. And one thing we can look at are which families are changing exceptionally fast. And since I'm not an expert on any of these particular species, I sort of asked the I5K community to look at particular nodes in the tree. And I think the most promising thing has come from Jessica Garb's group at UMass Lowell, which has been looking at the spiders group. And they found several rapidly evolving gene families involved in venom and silk production, which I think is really cool because it suggests that we're seeing genomic innovations that correspond to phenotypic observations. And I've been looking at a node of the German cockroach, Flotella germanica, because it has such a high number of rapidly evolving families. And I found one pretty interesting family that seems to be involved with response to light stimulus. And I think if anyone who has a lab in an older building is familiar with how cockroaches sort of scatter when you turn the lights on. So I thought that was pretty amazing. Another thing we can look at are gene families that are present on only a specific lineage or only within a group of species. So I took the order-specific gene families, and what I found was a high number of Lepidopteran-specific families. And I've been looking at the families, and I don't know why this is such a big peak. There doesn't appear to be any pattern here, but it is something interesting that I'm still going to be looking at. So now I want to shift from the overall patterns to focus on a particular point in time. And I want to focus on the ancestral insect, or as I call it, Leica. So when we are concerned with sort of the physical appearance of ancestral organisms, we have to rely on fossils. And there are some pretty good ones, like this ancestral mandibulate that was recently published. And these fossils can lead to some pretty fascinating artistic renditions of these organisms. But as far as the characteristics of the genome, how can we infer anything about those? Well, we can sort of use the same framework of gene families as before. We can take the observed counts of genes on the tips, and there are methods that we can use to infer the ancestral counts along the internal nodes. And then we can focus on a particular node, in this case the Leica node, the node ancestral to all insects, and we can ask some questions about reconstructions along that lineage. For instance, we can ask how many genes were present in the Leica genome. And what I'm doing is just summing up the total number of reconstructions at that node. When I do that, I get 9,601 genes. But actually, this is too small. It's biased because as we go farther back in time, there are more and more extinct families that we can't make any inferences about at all. So this number is too low. But since this is a simple sort of linear relationship here, we should be able to correct for it easily. And when we do that, we get a more reasonable number of 14,000 genes present in the ancestral insect. And that's represented by the orange dotted line on the left. So it's still a little bit of a small genome, but it's more in line with the sample. Perhaps a more interesting question we can ask are which families were born during the transition to insects. So then I'm looking for families that are represented by 0 counts outside of insects and are inferred as an expansion along the Leica lineage. And when I do this, I come up with 147 novel insect families, some of which have some pretty interesting implications for insect evolution. For instance, insects evolved changes in their exoskeleton, and we find 7 chitin cuticle production families that are novel to insects. Insects needed to evolve abilities to sense in the new terrestrial environment, and we find several sensory gene families that evolved along the Leica lineage. Insects also have unique developmental stages, with many of them undergoing a full metamorphosis. And we found 4 imaginal disc development families and 1 larval behavior family. One of the most interesting insect developments was flight, and we found 3 wing morphogenesis families that were novel to the insect lineage. So I hope to continue to be able to look at different points in time in arthropod evolution and reconstruct the genomes, maybe at the amino acid level. Until then, I've made this tool with all the gene family data available, and you can sort of click on a node and explore which families are evolving along your favorite lineage. So with that, I'd like to thank my advisor, Matt Hahn, Stephen Richards, and Rob Waterhouse for helping out a lot with the I5K, and everyone else in the I5K project as well. So thank you.
So if you move the Leica back to Datura, you get additional genes. What do you see in the origin of your genes? And
I haven't moved it back. yet. That's something I will do. I moved it forward to the holometabola node. There are actually no novel holometabola families, which I thought was kind of interesting.